Hello and welcome to this module on plasmids. This will be a brief introduction of plasmid replicons and associated concepts of replicons or two replicons. So, replicons or the term replicon is actually fundamental to understanding plasmids, more specifically to understanding the control of replication of plasmids. Now, when we discuss the control of replication with regards to plasmids, we are getting into areas that are actually quite nuanced and not fully understood. So you should keep that in mind as you go through this module. But this will be a good way to understand replication within the context of plasmids. So let me begin with a brief overview of plasmids. You are well aware that there are defining features of plasmids, and that includes that they are small and circular. So obviously here, small is a relative term. We mean that it is small relative to the chromosomal DNA of the host. In addition, plasmids are said to be extrachromosomal DNA. Now, this is just a way of highlighting the fact that the plasmid found in nature within a given host is not part of the genome of that host. Also, we say that plasmids engage in independent replication. And by that, we mean that the plasmid replicates independently of the host chromosome. Now, if you go or delve deeper into that and you ask, well, what do we mean by independence here? You'll find the term independence here is not very clear. So, when we talk about independence, generally in molecular biology, we sometimes talk about spatial or temporal independence. Spatial independence referring to two different things occurring in, say, different organelles, or temporal independence, whereby things occur at different times. So, in the case of plasmids, you will find that actually some plasmids are temporally independent from the replication of the host chromosomes, while others are not. So you have to keep that in mind as you go through this module. And I urge you to think about this question about the independent replication aspect. In addition, I'm sure you're aware that plasmids do rely on enzymes of the host to replicate. So you have to keep those things in mind. So in this context, independent replication simply means that the plasmid has its own origin of replication. Now, most commonly, plasmids are found in bacteria within nature, but in addition, they can also be found in archaea in some eukaryotic organisms like yeast, for example. We often discuss the fact that plasmids can carry genes that are beneficial to the organism. And a commonly remarked example of this would be antibiotic resistance genes, although you should recognize that there are other types of genes or other types of advantageous properties that the plasmid can confer onto the host organism. Plasmids are also dependent on enzymes of the host cell for things like replication and transcription. That is, the replication machinery of the host cell and the transcriptional machinery of the host cell will be used to replicate the plasmid and to transcribe genes within the plasmid. Now, in nature, lastly, plasmids can be transmitted from one organism to another. We see this through the process of conjugation, for example. This is the most common method by which a plasmid can be transferred onto another organism. Now, here to the right, you see a electron microscopy image of a plasmid. This is actually the first cloning vector used in 1973 by Stanley Cohen and Herbert Boyer. This is the PSC 101 plasmid, and it's helpful to remember this plasmid not only for its historical significance, but also because this plasmid utilizes initiator proteins, plasmid encoded initiator proteins, whereas other plasmids do not. So it's good to have some examples so that you can contrast different types of plasmids. So I'm sure you're aware of much of the information discussed here, and so I'll move on to the discussion of replicons. Okay, so before I go on, I do want to discuss some important terminology. So, first of all, when we think of the origin of replication, we often think of it as simply the DNA sequence at which replication begins. So, you may think of it as this region which will denature. That is, you have, say, this region within, say, the context of plasmids, where it is more prone to denaturing. That is, you can denature the two strands here. So a good generalization would be that it has more adenine thymine bases. And so we think of origin of replications as simply a region, a contiguous region, a small contiguous region by which replication can begin at. And this is actually a pretty good enough description or understanding of the origin of replication. So it's also an accurate understanding of the origin of replication. But if you go into the literature, you can actually get a little bit more of a nuanced way of looking at the origin of replication, but this will not be necessary in the context of understanding replicons. The case is not the same for the replicon. A replicon is a bit of a more abstract term, a bit of a more abstract concept, and you'll see that it has different word usages across literature. So, first, the replicon you can think of as a genetic unit that consists of the origin of replication. So, it consists of this, the origin of replication, but also all of its control elements. That is, all the control elements that are going to regulate the replication of the plasmid. Now, you have to be aware of the replicon's word usage. So, some will use the term replicon synonymously with the origin of replication. Replicon, more precisely, is the origin of replication, the replicon within the context of a plasmid. First, to the origin of replication, but also all the control elements. And this is fundamental. Otherwise, if you leave this part out, this whole discussion will be fruitless. Now, also in the context of a plasmid's replicon, you can also think of the replicon as the smallest piece of plasmid DNA, the smallest piece of plasmid DNA that can first replicate autonomously and also maintain normal copy number. Now, this is a very precise definition. There's a reason why they add those two things, that is, replicate autonomously and maintain normal copy number in this definition. This definition, more specifically, comes from this Sambrook book. It's a popular book called Molecular Cloning: A Laboratory Manual. But this is a very precise definition because, first of all, they say that a replicon is the smallest piece of plasmid DNA. That is, it is the region of the plasmid DNA, or it is simply the essential region of the plasmid DNA that is required for that region to replicate autonomously, replicate independently, but also maintain normal copy number. Now, the reason they add this here is because if you were to take this a small piece of plasmid DNA that can replicate autonomously, but say it does not maintain normal copy number of that particular replicon, then that means that you have left out some of the control elements. That is, you don't have all the control elements of that replicon, and thus you do not have the full replicon. So there is a level of abstractness here. However, if you keep those definitions in mind and wrap your head around them, then you should be fine. And so this is the basic idea of a replicon. I'll go into this in more detail later. Now, as I said earlier, the term replicon has some differing word usage, and this is why I want to add this term here, which is the essential region. It comes from this paper. This is actually a very excellent resource on understanding plasmid replication and the control of replication with regards to plasmids. And so in this paper, they say that in the plasmid, there's this essential region, quote unquote. And this is actually quite similar to the idea of the replicon, or to the discussion of a replicon. That's why I'm adding it here. But basically, the essential region would be the plasmid loci or genes that facilitate replication and the control of replication. So this would include the origin of replication, and it would include plasmid encoded genes that control replication. That is, they could be genes that produce RNAs that don't get translated into proteins, and they can control the replication of the plasmid, or they can produce proteins that control the replication of plasmid. So this is by necessity a vague discussion here. So remember, the term gene was classically used to discuss sequences that will produce a protein product. The term gene has evolved from that narrow definition. So in addition, plasmid encoded initiator protein would be part of the essential region, but you should recognize that this is contingent. So not all plasmids will have a plasmid encoded initiator protein. Now, just because a plasmid does not have a plasmid encoded initiator protein, that does not mean that that plasmid will not rely to some degree on a supplementary molecule, a supplementary initiator protein provided by the host cell. But the reason this is contingent is because some plasmids encode initiator proteins and others do not. I think this is clear. Now, the plasmid encoded genes will encode or express products that will regulate the replication of the plasmid. So is the case with the initiator proteins encoded by the plasmid. And so we'll call these regulatory molecules. They're regulating the control of replication. And what you should recognize is that depending on the properties of those regulatory molecules, we can make some generalizations as to how that will affect the behavior of replication of the plasmid. So we've discussed the replicon. We've also discussed the essential region, although it's not entirely the same as the replicon. So let me go ahead and discuss the replicon in more detail. First, the replicon, we said, is part, or in part, the origin of replication, but also all the associated control elements for that origin of replication. So I'll just say CE for control elements. Now, what you should recognize when you think of control elements is that there are regulatory molecules that will control the replication. We can divide them into RNA regulatory molecules and protein regulatory molecules. Recall, when I discussed the essential region, I discussed those regulatory molecules which are encoded by the plasmid. This is what I'm talking about here. So these can be proteins or they can be RNA. And now what you should recognize is that depending on whether they're RNA or protein, we can derive some generalizations as to the behavior of the replication of the plasmid. So for RNA molecules, you need to be thinking of positive and negative regulatory molecules. And for the protein regulatory molecules of replication, also you need to be thinking of positive and negative. If positive, then that will induce the replication of the plasmid. So it's a positive regulatory molecule of replication. It will induce replication of the plasmid. So we can just write induce replication. Now, importantly, if it is a negative regulatory molecule, then that will inhibit replication of the plasmid. And the same thing is the case for the protein regulatory molecules. If it's positive, it'll induce replication. If it's negative, it will inhibit replication of the plasmid. Now that we have these fundamental ideas down, we can further discuss other ideas pertaining to the replicon, but you should recognize that this is going to form the basis of your understanding for some of the basic concepts relating to replication of plasmids. So now that we have a general idea of replicons and what they are, let me discuss one of the fundamental things that replicons are responsible for, and that is the control of copy numbers for a given plasmid. So again, a replicon is a term that refers to the origin of replication and all its control elements. And copy number, you should recognize, refers to the average number of plasmid copies per host cell under normal growth conditions. So for example, if you have a particular host cell here and say its copy number is from 15 to 20, then we understand that on average within those host organisms that you have, say in a petri dish, you'll have 15 to 20 plasmids on average within each host organism. I will not draw the full number of plasmids, but that's the basic idea of the copy number. So that is the basic idea of the copy number. So it is a replicon that controls the copy number of a given plasmid. And more specifically, when we talk about the copy number, it is dependent on the plasmid's replicon in these ways. First, when we refer to whether the copy number is high or low, then this depends greatly on the positive control elements. More specifically, the positive regulatory molecules that regulate replication of the plasmid that I discussed earlier. These will influence copy numbers. So they could be RNA or they could be proteins. And depending on whether they're RNA or proteins, whether the positive regulatory molecules consist of RNA or protein will define the copy number. So on average, and this is a generalization, but it is a useful generalization, on average, a replicon that consists of positive RNA regulatory molecules will have high copy number versus a replicon that relies on positive protein regulatory molecules will have low copy numbers. Now, in addition, the negative control elements also help control the steady state of the copy number. So this is the state at which the number of plasmids within a given host cell remains relatively constant. So one model suggests that when you have the beginning of the colonization of a particular host cell with plasmids, say in a petri dish in a lab setting, what will happen is that once the characteristic copy number has been reached by that particular type of plasmid, then the negative control elements will increase in concentration within those plasmids, and it will suppress any further increase in the number of plasmids. So here to the right, you can see the PSC 101 plasmid, and you can see what is labeled a Rep 101 sequence. It sits right next to the origin of replication of this plasmid. And what you should recognize is that the Rep 101 sequence here is what will encode the Rep A protein. And the Rep A protein is a plasmid encoded initiator proteins. Now, not all plasmids will have a plasmid encoded initiator protein, but in this case, this plasmid does. Important to recognize here is that this is called the PSC 101 plasmid, but in addition, the replicon is also called the PSC 101 replicon. Not all plasmids will share the same name as a replicon, so you will find the PSC 101 replicon in other plasmids that are not called the PSC 101 plasmid, so keep that in mind. But basically, the point that I'm making here is just giving you an example of a plasmid encoded initiator protein that will help control replication that happens to be a positive regulatory molecule, and this would be the Rep A protein, the sequence of which is called Rep 101 here. So this would cause it so that the PSC 101 is, relatively speaking, result in low copy number. Now, an important thing to recognize is that replicons can be modified so that the copy number can be modified. We call it the copy number relies to a very large extent on the replicon. So example replicons that can be and have been modified to increase the copy number include the PMB1 replicon and the COL-E1 replicon. So both of these have copy numbers that are roughly between 15 to 20 in bacterial cells. And both the PMB1 and the COL-E1 replicons have been modified so that they increase the copy numbers. For example, there is the PUC plasmid. This contains a modified PMB1 replicon, and the copy number of this PUC plasmid is 500 to 700. So by modifying something within the replicon, something within the PMB1 replicon, we've increased the copy number from 15 to 20 to 500 to 700. This is a significant increase in the copy number, and it goes to show you how replicons influence copy numbers to a large extent. Importantly, you should recognize the difference between stringent and relaxed replication. So for instance, when we talk about relaxed replication, we're referring to plasmid replication that occurs independently of the host chromosomal DNA replication. In other words, here we're talking about temporal independence. That is, that the plasmids will replicate at a time that is not necessarily the same time at which the chromosomal DNA replication of the host will happen. Now, this happens, relaxed replication happens when the origin of replication is positively regulated by an RNA molecule. So the PSC 101 plasmid would not be an example of this, but the PMB1 and the CoLE would be. So remember, I introduced the CoLE1 in the previous slide and the PMB1 replicons, and these are fundamental replicons that are regulated by RNA positive regulatory molecules as well as RNA negative regulatory molecules. But when we talk about the relaxed replication versus the stringent replication, we are concerned with only the positive regulatory molecules. So the replication here does not rely on plasmid encoded proteins. It relies on plasmid encoded RNAs. Namely, you will find that these are RNA1 and RNA2. So these are replicons that have relaxed replication. They have relaxed replication because the origin of replication is positively regulated by an RNA molecule and not a protein. On the other hand, when we talk about stringent replication, referring to plasmid replication that occurs along with the host chromosomal DNA replication. That is, they are temporally dependent on the chromosomal replication of the host. And this happens when the origin of replication is positively regulated by proteins. And so an example of this would indeed be the PSC 101 replicon, which is found also within the PSC 101 plasmid that I showed you earlier. And so in this case, the PSC 101 plasmid would engage in stringent replication because its replicon has positive regulatory molecules that are proteins. And so this is why they engage in stringent replication. So there is a practical reason to understanding relaxed and stringent replication of plasmids. And one of those reasons is that you can use chloramphenicol so that you can perform relaxed plasmid amplification. So in the name, we're talking about amplification of plasmid DNA. And specifically, we're talking about those plasmids that engage in relaxed replication. So chloramphenicol amplification of plasmids, that is increasing the number of plasmids in a given host cell via chloramphenicol, only works if the plasmid engages in relaxed replication. It does not work if the plasmid engages in stringent replication. So keep that in mind. So basically, chloramphenicol is an antibiotic, and the mechanism here is what is relevant. Namely, chloramphenicol inhibits protein synthesis by binding to the 50S subunit of the 70S ribosome within the host. So because of this, what happens is that you have decreased protein synthesis. So let's say here to the right, you have a host cell in this big rectangle, and you add chloramphenicol here. So if you add chloramphenicol or you expose this organism or this host cell to chloramphenicol, all in all, you'll have decreased protein synthesis in the host cell. And once you have decreased protein synthesis, that results in decreased replication or diminished replication of the chromosomal DNA of the host cell. Now, if you get into the details here and try to understand it in detail, you'll realize that this can be nuanced and it's not as simple as I am describing it. But one way to look at this is that the host cell, to replicate the chromosomal DNA, the host DNA, requires certain proteins to be present at every S phase of the cell cycle where you want to replicate the host DNA. So there are certain proteins that require continuous replication with each cycle. Now, in the case of the decrease of protein synthesis, you'll still have those major enzymes that are part of the replication machinery, but you will not have the proteins, say the initiator proteins that initiate replication of the host DNA. This is one way to look at it, although I must admit it does not provide the full picture. This is more nuanced than simply that. So this has an effect on the plasmids. So if the plasmids engage in relaxed DNA replication, so for example, if all these plasmids are either the PMB have the PMB1 replicons or have the COL-E1 replicons, they cannot have you cannot have a mixture here because these would be incompatible. But if you have all plasmids that have the PMB1 replicons or all plasmids here that have the COL-E1 replicons, then this would mean that these engage in relaxed replication. Also means that they do not rely on plasmid encoded initiator proteins. Consequentially, you are not relying on protein synthesis as much to replicate those plasmids. And because of this, you'll see that the relative content of the number of plasmids will increase after you add the chloramphenicol relative to the content of the host DNA. In fact, after mere hours of chloramphenicol exposure, the relaxed plasmid DNA content will increase rapidly relative to the host DNA content. So a simple way, as I described, to understand this is that, simply speaking, in the case of relaxed plasmids, the functional protein synthesis of the host organism is relatively more important for initiating the chromosomal replication of the host or the replication of the host DNA than it is for replicating the relaxed plasmids. Importantly, you should recognize some aspects about the positive regulatory molecule in the context of replicons. And you should recognize the differences that entail by having either RNA positive regulatory molecules or protein positive regulatory molecules. So recall that the plasmid replicon controls the copy number, and replicons with RNA positive regulatory elements tend to have higher copy numbers. They replicate in a relaxed fashion, as I mentioned earlier. And also, you can have increased plasmid DNA to host DNA ratio with the addition of chloramphenicol, or alternatively, amino acid starvation, adding a condition whereby you starve your host cells from amino acids. Now, as I said earlier, both the PMB1 and the COL-E1 replicons have RNA positive regulatory elements. In fact, they have positive and negative regulatory molecules, with the RNA2 being the positive regulatory element or molecule and the RNA1 being the negative regulatory part. So before I go on, let me make a quick clarification here. So when I say RNA2 or RNA1 regulatory molecules, I'm talking about the expressed RNA transcripts that regulate the replication of the plasmid or control it. But when I say an RNA2 or RNA1 element, then I'm talking about specifically a sequence of DNA within the plasmid that is going to transcribe the RNA2 and RNA1 regulatory molecules. So in this case, the RNA2 and 1 regulatory molecules would be named the same as the RNA2 and RNA1 regulatory elements or control elements. And it would be pivotal to understand the actual mechanism of RNA2 and RNA1 in the context of plasmids, because these are utilized within the PMB1 COL-E1 replicons, which are very widely used in plasmids. So a lot of plasmids use the PMB1 replicon, for example, or a derivative of the PMB1 replicon. Now, on the other hand, replicons with protein positive regulatory elements replicate in a stringent fashion. Because of this, they tend to have low copy numbers. In addition, they also replicate in a stringent fashion, and also chloramphenicol and the condition of amino acid starvation both cannot amplify plasmids that contain those types of replicons. So the PSC101 plasmid or PSC101 replicon would be an example of a replicon that has a positive regulatory molecule or element that is a protein. Here to the right is again the PSC101 plasmid with the PSC101 replicon, and this replicon would consist of the PSC101 origin of replication, as well as all the control elements, which include the rep 101. So I do want to briefly discuss the control of copy numbers with regards to plasmids. So you should recognize that at steady state, the plasmid's rate of replication on average is 1 replication per plasmid and host cell cycle. Now you should recognize that to reach that characteristic copy number, there has to be a certain regulatory means by which that characteristic copy number is first of all reached and then maintained. And so this is an area that's under research, but there are some models that have been described to try to explain the way by which a plasmid reaches the characteristic copy number and maintains it. So one way to think of this is basically as follows. When you have the initial plasmid colonization of the host, you have a decreased concentration of the negative regulators. And because of this, because you have less of those negative regulators, then that permits the characteristic copy number to be reached. In other words, you're not hindering the replication of the plasmids, and you're allowing those plasmids to replicate to reach that characteristic copy number. Now, as or after the copy number is reached, those negative regulators will increase in concentration or in numbers. And so, because of this, you now have some kind of a halt to the rapid increase in the plasmid or the rapid replication of the plasmids that was occurring during the initial colonization. And because of this, now you have this characteristic copy number that's been somewhat reached. Now, importantly, the rate of replication will then increase or decrease as necessary to maintain the copy number. And this is where the real puzzle occurs. This means that you have mechanisms by which the plasmid concentration is measured. So, because of this, you should recognize that there are concepts pertaining to the control of plasmid replication that continues to be elusive. Now, relating to the topic of plasmid replicons is incompatibility. So, when we talk about incompatibility in the context of plasmids, then we're talking about a scenario whereby core resident plasmids cannot be stably maintained without some external selection. So, different plasmids with replicons of the same negative regulatory elements tend to compete inside the host cell. Now, this is important to recognize because we're talking about different plasmids with replicons that utilize the very same negative regulatory elements. So they do not have to be the same replicons, but they can be replicons of the same negative regulatory elements or replicons that utilize the same negative regulatory elements. And this would make such plasmids that utilize those replicons with the same negative regulatory elements incompatible with one another. So, in such a scenario, we call this incompatibility, and such plasmids cannot be maintained in the same host cell. Now, you should recognize that different plasmids with replicons of differing negative regulatory elements do not compete inside the host cell. And because of this, such plasmids can be maintained inside the same host cell. So, here to the right is a figure. You can see different plasmids with the replicons of those plasmids, as well as the copy number of those plasmids. Now, as I said earlier, the copy number is largely determined by the replicon. So, this should be clear by now. So in this figure, you should probably recognize those replicons right here, the CoLE1 and the PMB1 replicons. As I said earlier, the CoLE1 and the PMB1 replicons in a bacterial host under the appropriate growth conditions will have copy numbers of roughly 15 to 20. So from this, you can probably recognize that this PMB1 replicon is modified, and this is why you have a very high copy number for the PUC plasmid. You should also recognize PSC 101 replicons here. The PSC 101 replicons are replicons that utilize a plasmid or that are associated with plasmid encoded initiator protein, namely the Rep A protein that I've discussed earlier. Because of this, this engages in stringent plasmid replication. Because of this, it has a low copy number. Remember, these are generalizations, but they are helpful generalizations. So the copy number of the PSC 101 replicon is close to 5. So from this, you should probably recognize that this PSC 101 replicon is a derivative, or it's a modified PSC 101 replicon because it has a high copy number here. So the basic idea of incompatibility is that if you have different plasmids that are co-residents within the same host cells, then those plasmids, if they utilize replicons with the same negative regulatory elements, would be incompatible. So in the case of those plasmids right here that you see in blue, they all utilize the CoLE1 and the PMB1 replicons, which use the same negative regulatory elements. And because of this, those would be incompatible replicons, and those would be incompatible plasmids. The same is the case with all of these plasmids that you see here. If they're color coded with the same color, that means that they are incompatible with one another. Here you can see that those plasmids with the PSC 101 replicons would be incompatible with one another. Now you can add a PSC 101 plasmid with a PSC or a plasmid that utilizes the PSC 101 replicon with a plasmid that utilize a CoLE1 or a PMB1 replicon, for example, because they rely on different negative regulatory elements. Now, in addition, on this figure, you can see other types of replicons that I have not discussed here. I will discuss them later on, hopefully. But you can see, for example, the F1 and the P1 replicons here. Those are derived from bacteriophages or animal viruses. They can be helpful in certain circumstances, which is why I'll discuss them in a later module. Importantly, if there's one thing that you should recognize in the slide, is that incompatibility is caused by the replicons of two different plasmids having the same negative regulatory elements. Importantly, those are generalizations. And so when you use generalizations, you should always have a level of doubt in your head about whether that generalization applies or not. But they are useful generalizations and they do work pretty well for the plasmids that are prevalent or contemporarily used in laboratory research. There is another type of incompatibility caused by problems in partitioning or the segregation of plasmids because of the inherent partitioning systems that are found within plasmids. So, very briefly here, when we talk about partitioning or segregation within the context of plasmids, we're referring to the mechanism by which plasmids are partitioned to the host daughter cells. So, low copy number plasmids in particular depend significantly on the partition systems for them to be stably segregated into daughter cells. So, you should recognize that there are partition systems in plasmids, and those are inherent to the plasmid. Specifically, the partition systems, or most partition systems, are encoded by the plasmid. They're encoded by an area of the plasmid which are called the par operons. You're very well aware of what an operon is, so I'll not discuss this here. Now, if you want to delve deeper into this, you can. This is a very useful paper to look into the partitioning system. For example, this paper discusses the significance of probability and the differing influence of the incompatibility problem due to partitioning in low copy number versus high copy number plasmids. Namely, when we're dealing with high copy number plasmids, what happens is that the partitioning relies greatly or more greatly on probability or on randomness of the separation of the plasmids. However, when you're dealing with low copy number plasmids, you do have to rely more heavily on those partition systems that are encoded by the par operons of the plasmids. Now, when it comes to the partitioning systems of plasmids, the nucleotide triphosphatase and the centromere binding proteins are going to be central components that are responsible for partitioning the plasmids that use partitioning systems. You can look at the paper below, namely the first one cited, to learn more about partitioning systems. So, very briefly here, I do want to summarize much of the central concepts discussed in this module. First, we said that a replicon is a term that has different word usages. And so I defined the replicon as to how I'm going to use it in this module. I also suggested that you look into what is called the essential region as a term or a way of thinking of the similar concepts that are derived or that we can understand by understanding replicons. The problem with using the central region as a term or as a way or as a concept is that it is not widely prevalent. So we do have to, by necessity, utilize the term replicon. And so when it comes to replicons, you should recognize that they consist of the origin of replication as well as the control elements. And I said that when you think of the control elements, always think of the regulatory molecules and whether they are RNA or protein. And if they are RNA or protein, that will determine the behavior of the replication of plasmids to some degree. So, in the case of RNA regulatory molecules, they could be positive or negative. And in the case of protein regulatory molecules, they could be positive or negative. And you should recognize what kind of information you can derive by knowing the positive regulatory molecules used by a given plasmid or at a given replicon. So, in the case of RNA positive regulatory molecules, those would lead to relaxed replication of the plasmid. And in the case of a positive protein regulatory molecule, this would lead to stringent replication of the plasmid. We also said that the relaxed replication or the plasmid that engage in relaxed replication can be amplified using chloramphenicol or amino acid starvation conditions. In addition, I also said that when you have positive RNA regulatory molecules, this tend to result in high copy numbers. And when we have positive protein regulatory molecules, this tends to result in low copy numbers. Now, in addition, I also said that incompatibility can be due to having co-resident plasmids that utilize the same negative regulatory elements. So if you have two different plasmids that utilize the same negative regulatory elements, say the same negative RNA regulatory element or the same negative protein regulatory elements, then this would make it so that you have incompatibility of those co-resident plasmids. This concludes this very brief introduction on replicons and associated concepts.